Out of an initial deployment of 300 million sperm cells, only a microscopic fraction will survive the journey to their final destination. Early thinkers looked at reproduction and assumed male fluid contained fully preformed miniature humans that simply grew larger over time. When early microscopes first revealed the existence of swimming sperm cells in the 1600s, scientists falsely concluded that the male provided the entire human form, while the female merely acted as a passive incubator. That assumption stood until 1827, when a scientist named Carl Ernst von Baer finally observed a mammalian ovum, or egg cell. This proved the female makes an equal biological contribution to the new organism. We now know fertilization is an intense biological elimination tournament between two highly specialized gametes, designed by evolution to destroy any signs of weakness. The gauntlet begins immediately. The vaginal canal is highly acidic, operating at a pH level around 3.8. Within minutes of entry, millions of sperm are dissolved by this hostile environment. Those that survive face a physical wall, the cervix, which remains tightly closed to block entry for the vast majority of a woman's menstrual cycle. Only during ovulation does the thick cervical mucus thin out to a more watery consistency. Even then, it forms a microscopic labyrinth that traps malformed cells and permits only the most structurally sound sperm to pass. Inside the uterus, the survivors are hunted. Resident white blood cells mistake the sperm for foreign pathogens and consume thousands of them on sight. The female reproductive tract establishes these immediate lethal environmental barriers to actively select for viability, ruthlessly testing the physical endurance of every single cell. To push deeper into the reproductive tract, the surviving sperm require a biochemical upgrade known as capacitation. Reproductive fluids strip away cholesterol molecules embedded in the sperm's outer membrane. This physically thins the membrane and triggers a state of hyperactive swimming. By the time they enter the fallopian tubes, only a few thousand contenders remain. Here, they must surge upstream, fighting against thousands of tiny hair-like cilia that constantly push fluid in the opposite direction. This visualization shows how the egg actively guides the sperm. Surrounding cells release progesterone, creating a chemical gradient that intensifies closer to the source. The hyperactive sperm detect this gradient, aligning themselves toward the highest chemical density to successfully locate the egg. Reaching the egg relies on a precision molecular targeting system that requires both an environmental upgrade and complex chemical signaling. The final few dozen sperm arrive to find a massive cell protected by two heavy defenses: an outer layer of follicle cells called the corona radiata, and a thick protein shell called the zona pellucida. To get through the shell, a sperm must successfully bind to specific glycoprotein receptors covering the surface of the zona pellucida. This receptor binding is the exact trigger for a necessary mechanism called the acrosome reaction. The cap of the sperm bursts, releasing digestive enzymes. These chemicals literally dissolve a physical tunnel through the egg's dense extracellular matrix. Successful penetration depends entirely on this molecular compatibility. These specific protein interactions prevent cross-species fertilization and ensure that only cells with the correct chemical profile can successfully enter. The instant the first sperm makes contact with the inner plasma membrane, the egg physically pulls the successful cell inside and initiates a rapid lockdown. This speed is critical to prevent polyspermy. If multiple sperm were to fertilize a single egg, the resulting organism would have extra sets of chromosomes, making it biologically fatal. To guarantee only one sperm gets in, the egg deploys a definitive secondary defense mechanism, known as the cortical reaction. This heat map visualization demonstrates how a wave of intracellular calcium triggers the release of cortical granules. This instantly hardens the zona pellucida into a rigid wall, forcibly ejecting all other competing sperm. The egg acts as a highly secure vault. Rather than sitting as a passive finish line, it actively seals its perimeter the millisecond it is breached. Inside the vault, the sperm sheds its tail. All that remains is its genetic payload and a crucial biological engine called a centriole. The newly introduced centriole casts out a spiderweb-like structure of microtubules, physically reeling the male and female pronuclei toward each other. The two sets of 23 chromosomes fuse. In an instant, they combine to create a completely unique human genome that determines traits like eye color, hair color, and body structure. This new single diploid cell is called a zygote, and it contains the complete operating instructions for a human body. After a hostile journey that destroyed hundreds of millions of cells, this singular moment of genetic fusion marks the definitive end of the biological gauntlet and the exact beginning of human embryogenesis.